and get some housekeeping out of the way. So if you refer to your syllabus schedule, you'll notice that next week is exam 1. So make sure you're not waiting till the last minute to study for this exam. It will cover chapters 1, 2, and 4. I anticipate we'll finish chapter 4 by Thursday's class. Yes? Yeah, that's very helpful, absolutely. For the exam, it'll be next Thursday, so that means that you'll have all the material at least a week ahead of time, right? Because whatever we cover on Tuesday's class will not be on the exam likely, assuming we finish chapter 4 on Thursday. So that gives you guys extra time to study. We are going to go back to chapter 3, but that'll be after exam 1. The format for the exam will include multiple choice, but I also plan to have some fill in the blank and short answer questions on there as well. Given that this is an upper level course and it's not that big of a course, then I think I can handle grading some short answer. And I think it's good for you guys to be able to explain how you're thinking when given a particular question. It also opens up the opportunity for partial credit, which is good. Multiple choice, you can't really give partial credit. Yeah.
So for short answers, would you like to do a paragraph?
Oh, I'll give you instructions on the test. Yes, they'll be very specific. Don't worry about that. So you don't need to bring a Scantron or anything. You'll just answer the questions on the test, and I'll collect those and grade those. Okay? Also, if you have any questions specific to the test, you can always email me or you can come and see me during office hours. You have the full class period to complete the exam, 1 hour and 15 minutes. So make sure you're here that day. There are no makeup exams. So absolutely need to be here, okay? Now, if you have a university approved excuse with documentation, then yes, you'll be able to make up the exam. But other than that, you need to be here. You need to be here on time. Sometimes students will come in late, and that's a problem. So you'll be denied the exam if you come later than the first person's turned it in. Sometimes I have students turning in exams pretty quick, like within 20, 30 minutes. And if that happens and you come in and the student has already turned it in, then you won't be able to take the test. It'll be a 0, okay? So you have to be here on time. Because students would talk to the person outside, get answers, and then come in. I've seen a lot over the past 10 years. So there's reasons for everything that I do. Also, no baseball caps, please, during the exam, or you turn it backwards. Students are very creative with their cheating, I've noticed over the years, okay? No smartwatches, no cell phones on you. That's just about all the things. And then I do have your next quiz available. Did you guys see it? Quiz 2 should have opened up about 10 minutes ago on Blackboard. It covers chapter 4. So quiz 1 covered chapters 1 and 2. Quiz 2 covers chapter 4, which is the subject of today's class and Thursday's class, if we can actually have class. It's due Friday at 5. Did anyone have any trouble with turning in the online quiz last week? I'll know more about the test after I've started writing it. So stay tuned on Thursday. When you get a class right before the semester starts, you're going just one class ahead of the students. And I have that for 2 of them, so please be patient with me. Well, he said he was going to hurry. All right. Sorry about that, guys. Oh, well, you got out of 30 minutes of micro. Don't be too happy. All right, better late than never. Okay, so what we're going to do today is we're going to finish up chapter 2. We don't have much to go on that, okay? And then we're going to jump into chapter 4, right, which talks about the nutrition and growth requirements for microorganisms. Then after exam 1, we'll come back to metabolism, which is chapter 3. So I'm going to turn off the lights. I found what I think is a cool video. All about flagella. Who can remind the class what flagella are for? Movement, motility, that's right. So we talked about how in bacteria you have this sort of tumble and run effect based on the flagella moving counterclockwise. They'll move together, and so the flagella will run, or the bacteria will run. But then if it goes in the opposite direction, say clockwise, then they start to spread apart, and you get this kind of tumbling action, right? So today we're going to continue the discussion of flagella, and we're also going to get into some growth requirements for bacteria. So let's watch this video, and then we'll pick up the lecture.
Many species of bacteria propel themselves through their environment by spinning helical motorized flagella. Rotobacter cells have one flagellum each, whereas E. coli cells have multiple flagella that rotate in bundles. Each flagellum consists of a helical filament that is 20 nanometers wide and up to 15 microns long, and spins on the order of 100 times per second. These animations show a series of schematized and speculative models about how bacterial flagella might function and assemble. Just outside of the cell wall, the filament is connected to a flexible rotating hook. The filament, the hook, and a structure called the basal body, located below the cell's surface, make up the three parts of the flagellum. The basal body consists of a rod and a series of rings embedded in the inner membrane, the peptidoglycan layer, and the outer membrane. Some of the rings make up the flagellar motor, which can be divided into two major parts: the stator, which is attached to the peptidoglycan layer, and as its name implies, remains stationary, and the rotor, which rotates. The motor derives its power from a proton gradient across the membrane. In this example, a high concentration of protons exists outside, and a low concentration exists inside the cell. The protons flow through the interface between two types of proteins, called mote A and mote B, that make up the stator. Mutational studies suggest that a conserved aspartic acid in mote B functions in proton conductance. Each stator contains two mote B proteins, and therefore also contains two of these important aspartic acids. Although the molecular mechanism of rotation is not known, one possible model describes protons moving through the channels in the stators and binding to the aspartic acid in the mote B proteins. This binding causes a conformational change in mote A proteins, resulting in the first power stroke that moves the rotor incrementally. At the end of the first power stroke, the two protons are released into the cytoplasm. The proton loss causes a second conformational change that drives the second power stroke, once again engaging the rotor. Although the mechanism for motor function is not yet certain, many details of flagellar assembly have been determined. Flagella begin their assembly with structures in the inner membrane. 26 subunits of an integral membrane protein called Fle-F come together in the plasma membrane to form the MS ring. The Fle-G proteins assemble under the MS ring. Fle-G, along with Fle-M and Fle-N proteins, make up the rotor. Flagellar proteins destined to be part of the extracellular portion of the flagellum are exported from the cell by a flagellum-specific export pathway and assembled at the center. Mote A and mote B form the stationary part of the flagellar motor, the stator. Both are integral membrane proteins, but mote B is also anchored to the rigid peptidoglycan layer, keeping the stator proteins fixed in place. The subunits of the rod portion of the rotor move up through the hollow cylinder in the assembly, and assisted by cap proteins, build up the rod in a proximal to distal fashion. Another set of rings, called L and P rings, are found in gram-negative bacteria such as E. coli. They penetrate the outer membrane, forming a bearing for the rod. As the rod cap is exposed outside the L ring, it dissociates and is replaced by a hook cap that guides the assembly of the hook proteins. After the hook is assembled, the hook cap dissociates, and a series of junction proteins assemble between the hook and future filaments. Finally, yet another cap is built, and filament proteins assemble. Like the rod and hook proteins, they travel through the hollow channel inside the filament to reach the distal end.
It's mesmerizing.
The cap rotates, which causes the subunits to build in a helical fashion. A complete filament can consist of 20,000 to 30,000 subunits.
One of the great challenges in this world is knowing enough about.
All right. So just as we learned in the video, guys, as far as the structure of the flagellum, it is rigid and it's considered helical in shape, and it's made up of several components. Right? So you have the part that's more stationary and embedded in that membrane material. And then you have the part that is moving, right? That allows for this sort of rotation. So the main part is called the filament. It's composed of flagellin protein. You also have the hook portion here, and then the motor, which makes up the basal body. And it's a rotating machine. So several genes are required, as you saw in the video. The MS ring is the first thing that's made. You have anchoring proteins, the hook, the cap, they're all made next. And then the filament grows from the top, which was the last part of the video, which was really cool to watch. And the flagellum gets exported through that export apparatus. So I'd like you guys to know in general how the flagella are made and how they work, right? And their significance for bacteria. Now, archaea also have similar flagellar structures that help in their movement, and they're called archella. These proteins are unrelated to flagella. They're more closely related to a different structure on bacteria known as the pili, type 4 pili. But nonetheless, they do share some features with flagella, and they allow for movement. They're considered smaller than bacterial flagella as a whole, only about 10 to 13 nanometers. And they do move by rotation, driven by the hydrolysis of ATP. As far as the movement or the motion, it is helpful, but it's not as efficient as the movement that bacteria flagella provide. So they generally will swim much more slowly. There is an exception for one of the methanogens shown here. And this one will be able to move a bit.
So they're powered by the hydrolysis, and the flagella are powered by the proton.
Yes. Some cool EM images shown here too. All right. Now let's talk about this movement. It typically will occur away from the colony of microorganisms. It's slower than swimming, and there's either twitching or gliding motility that can happen. So here you have a bacterial cell, rod-shaped, with a pilus coming off of it. It will attach to the surface and then pull the cell forward. This is a twitching motion. This is what we call surface motility, guys. So you're not using the flagella to rotate. You're using a structure that attaches to a surface to allow for movement. So here's your rod-shaped, bacillus-shaped bacterium. Here's your pilus. It attaches to the surface, and then it pulls the cell forward, right? Cell moves in this direction. There's also a gliding motion that can occur in the bottom part of the figure. Here you have these adhesion proteins surrounding the cells, and they allow for the gliding over surfaces as the cell rotates over that surface. So suffice it to say that microorganisms do have multiple ways by which they can move, flagella being sort of the top dog, the one that's mostly used and most efficient, but there are other ways as well, such as the surface motility that we're seeing with gliding and twitching. Now, why would a microorganism want to move? Well, it makes sense if they live in a liquid environment, right, to acquire nutrients or to get to safety, whatever it is. So when we have this directed movement, we have a name for it, we call it taxis. And this is directed movement in response to some sort of stimulus. And that stimulus can be chemical or it could be physical. In the case of chemical response, it's chemotaxis. Chemotaxis, you've probably heard this. So maybe a particular microorganism senses a chemical, and maybe that chemical is beneficial to it, right? It's some sort of mineral maybe that it needs, it's depleted of. And so there will be directed movement toward that particular chemical, so attraction to that chemical. Or maybe it senses some sort of danger in the form of a chemical that it doesn't want to come near because it could harm it or kill it, right? In that case, it would be directed movement away from that stimulus, obviously. So chemotaxis would be the attraction or repellent of a chemical, and therefore the response that ensues. But you have other types of taxis as well, in addition to chemotaxis. You have phototaxis. When you hear photo, what do you think of? Light, right? So attraction to or repulsion from light. You have to remember it goes both ways, right? It's not always movement toward light. It could also be movement away from light, depending on the situation that that microorganism is in. So this directed movement is going to enhance access to resources and allow these organisms to avoid things that they don't want to be near because it could cause them damage or death. All right, and we see this, guys, this taxis in both bacteria and archaea. And so here is a picture from your textbook which shows you chemotaxis in flagellated bacterium. On the left, no net movement. These guys are tumbling and running and tumbling and running, right? So again, when those flagella are working in concert together due to the clockwise or counterclockwise motion of the flagellum, then it's able to move in one direction. But then the minute those flagellum spread apart due to a reversal in the rotation, then you get this tumbling in all different directions, right? Until they come together again and act as one to move in one direction. So this tumble and run is characteristic of what we see bacteria do when we look at them under a microscope. And then if an attractant or a repellent is present, then we can see directed movement toward or away from that attractant or repellent. In addition to chemotaxis and phototaxis, there are some others, and I'm not going to go into great detail about, but your book does, so you're welcome to read about it. But guys, if I don't go into detail on something in lecture, that means I'm not going to ask you about it in detail on the test, okay? When you're studying for my test, let the lectures be your guide. If I talked about it in lecture, yes, you need to know everything about it, okay? And that's what I'm going to ask you about, but I will not ask you about things that I did not cover in lecture, okay? So let the lectures be your guide, and then read the appropriate parts of the textbook to supplement your knowledge. So there's osmotaxis. I want you guys just to be aware of that term. That's in response to ionic strength. There's hydrotaxis. Hydro meaning water, so response to water. Aerotaxis, response to oxygen. And we already talked about phototaxis, response to light. All right, so for phototaxis, this is one that I do want to go into a little bit of depth about. This is when you have attraction to light. This allows for those organisms that are phototrophic. What does it mean to be phototrophic? Food, right? Chemical energy from light energy, right? Able to use the energy from the sun in order to make their own food. So these phototrophic organisms can optimize themselves in such a position to receive the most amount of light energy so that they can make the most amount of chemical energy to fuel their functions, right? Scotophobotaxis, it's a long one. Entering darkness causes the cell to tumble, heads back to the light via a biased random walk. So there's a particular type of movement associated with phototaxis. And then the photoreceptor is the protein that acts as a sensor that's going to initiate this process, sense the light, and then get the organism flagella to go and rotate in that direction toward the light. And so I wanted to show you, I do have one more video. I didn't realize it'd be so hard to watch videos today. I want to show you one more video about chemotaxis. No, it's not. I was so proud of the videos I've done. All right, one more video.
E. coli and a variety of other bacteria use flagella to swim toward chemical attractants, such as amino acids and carbohydrates present in media. The bacteria use a mechanism called chemotaxis, in which they sense chemical gradients and modify their motility in response. The flagella rotor can rotate clockwise or counterclockwise. In the counterclockwise mode, all flagella sweep behind the cell, forming a rotating bundle that propels the organism straight ahead in what is called smooth swimming or a run. If the flagella rotors suddenly switch to a clockwise rotation, the bundle is disrupted and the bacterium tumbles in a random fashion. Once the rotors switch back to a counterclockwise rotation, the bacterium will have reoriented itself, changing its swimming direction. The natural bias of a flagellar rotor is to turn in a counterclockwise rotation. However, the flagellum switches directions randomly at a particular rate, which is why the cell periodically tumbles. The key to chemotaxis is suppressing the number of tumbles as a cell moves from a lower concentration of a chemical attractant toward a higher concentration. The switching is minimized if the cell disables enzymes called Ki-A kinase. Ki-A kinase is found attached to a protein called Ki-W, which acts as an intermediary protein between Ki-A and a chemoreceptor called a methyl-accepting chemotaxis protein, or MCP. When an attractant binds to the receptor, the cytoplasmic domain of the receptor changes conformation. The conformational change thereby inactivates Ki-A. In this state, the cell is detecting a chemical attractant, deactivating Ki-A, and maintaining its straight-ahead run. In contrast, when no attractant binds to the receptor, Ki-A becomes activated. Activated Ki-A uses ATP to phosphorylate itself, and then it transfers the phosphate groups to a protein called Ki-Y, which regulates flagellar rotation. A few other proteins called Ki-B and Ki-R participate in sensory adaptation, but we will ignore them for now. When Ki-Y is phosphorylated, it binds to the rotor of the flagellum, reversing its rotation to clockwise. The cell tumbles and then faces a new direction in which to possibly encounter the chemical attractant. Thus, when no attractant is bound, Ki-A is active, Ki-Y is phosphorylated, and the cell tumbles frequently. To keep the response dynamic, the stimulus is quickly terminated by the action of a protein called Ki-Z, which removes the phosphate from Ki-Y. Dephosphorylated Ki-Y cannot bind to the rotor, and the flagellum goes back to its normal, smooth-swimming counterclockwise rotation, now propelling the cell in a new direction. Although we've described the action of each protein as if only one exists in the cell actually contains many molecules of each. As shown in this computer-generated image of E. coli, MCP sensory proteins assemble as clusters at the poles of the cell. A cluster at one pole is depicted here. Flagellar rotors are shown as blue and purple. Ki-Y proteins are shown as black dots, while the phosphorylated versions of Ki-Y are red. A high rate of tumbling depends on a high concentration of phosphorylated Ki-Y. Let's look at the process again, this time considering a phenomenon called adaptation, which involves the proteins Ki-B and Ki-R. Because the MCPs act more or less in unison, for simplicity, we will show just one MCP here. When no attractant is present, Ki-A is active and phosphorylated. Ki-A phosphorylates both Ki-Y and Ki-B. The Ki-Y and Ki-B proteins undergo continual turnover, losing their phosphates spontaneously, or in the case of Ki-Y, by the action of Ki-Z. Ki-Y and Ki-B are phosphorylated by Ki-A as part of a continual cycle. A high level of phosphorylated Ki-Y results in a high frequency of tumbling. If attractant is added to the environment, the attractant binds to the receptor, causing the inactivation of the bound Ki-A proteins. Ki-A gives its residual phosphate groups to Ki-B and Ki-Y. However, after a while, Ki-B and Ki-Y lose their phosphate groups and become inactive, and they no longer have Ki-A available to phosphorylate them again. With fewer phosphorylated Ki-Y proteins, the cell tumbles less often, swimming straight ahead while sensing the chemical attractant. It's advantageous for the cell to move to a higher concentration of the attractant. As part of a sensory adaptation mechanism, the cell becomes adapted to the current concentration of attractant by the action of key R. Key R slowly adds methyl groups to receptors, preferentially methylating attractant-bound receptors. As the struts of a receptor become methylated, they come together, returning the key A proteins to an active state. Activated key A can again phosphorylate key Y and key B. As key Y proteins become phosphorylated, they are able to bind to the rotor. The cell returns to a high rate of tumbling, even though it is in the presence of attractant. The cell has adapted, and its tumbling provides the cell with an opportunity to swim into an area with a higher attractant level. With the higher concentration, more attractant molecules are likely to bind the receptors, and when they do, they trigger the deactivation of the associated key A proteins. As a result of less key A activity, key B and key Y are also phosphorylated less. With fewer phosphorylated key Y proteins, the cell tumbles less and swims straight ahead more. Again, key R adds methyl groups to the struts of the receptors. The heavily methylated struts come back together, allowing the key A proteins to become activated again. The number of phosphorylated key B and key Y proteins increases. Thus, the cell adapts to the new higher level of attractant, and the cell returns to a high rate of tumbling. How does a cell become sensitive to attractant again if it is reoriented to swim away from attractant? The critical molecule is key B. The phosphorylated form of key B opposes the activity of key R by clipping off the methyl groups that key R has added. If a cell tumbles into an area of little attractant, the receptor eventually gets reset by losing its methyl groups to key B activity. When attractant is removed, the receptors are not methylated as much because key R prefers to methylate receptors that are bound by attractant molecules. Key A kinase is active and phosphorylates key Y, which will increase the frequency of tumbling and the likelihood the cell will reorient to encounter attractant. Unmethylated MCPs are again sensitive to attractant binding, which will again suppress the frequency of tumbling events so the cell can continue to swim into the attractant. The result of changing the frequency of tumbling as the cell moves away from attractant, increasing the frequency of tumbles, or toward an attractant, decreasing the frequency of tumbles, means the net movement of the cell will be toward higher and higher concentrations of attractant. Because attractants typically indicate a food source is nearby, chemotaxis is an important mechanism for survival in the natural environment.
That video went into a great deal of biochemistry and pathways. So you are not responsible for knowing all of those key proteins, CAG proteins. But hopefully you can appreciate after seeing that how much research has gone into the tumble and run of bacteria. I mean, all of that whole system that you just saw is probably described in like 100 different papers. I mean, it's pretty amazing how much we know about how that works. It's a very active area. All right, the last part of chapter 2, guys, is actually on eukaryotic microorganisms. These guys have organelles, as you know, because that's what separates eukaryotes from prokaryotes, right? They have a nucleus, which is membrane-bound, and other membrane-bound organelles. So let's talk about the structure of eukaryotic microorganisms. So they have this nucleus in the middle, this giant green sphere in the middle of the cell. That's going to, of course, house the genetic component, DNA. And then surrounding it, you're going to have your endoplasmic reticulum, rough and smooth. You're going to have mitochondria. What's mitochondria for?
Powerhouse of the cell.
Yeah, powerhouse of the cell, energy production, right? You also have lysosomes, you have Golgi, you have microfilaments, microtubules, all these different kinds of membrane-bound organelles that have specialized features, right? So all of these different structures that we see in eukaryotes allow for compartmentalization of the eukaryotic cells so that it can become better at doing certain tasks. And that's why eukaryotes are considered higher order organisms compared to prokaryotic microorganisms. Some of them even have additional organelles, such as the chloroplast, if it's a photosynthetic organism, and then our friends flagella and cilia for movement, such as in the case of eukaryotic sperm cells, and cell walls, which we've talked about a lot, that's present on microorganisms that are prokaryotic. You also have some cell walls protecting eukaryotic microorganisms. The membranes of eukaryotes also contain sterols. You'll recall those are derivatives of steroids, such as cholesterol, right? And this is going to be embedded between the phospholipids of the membranes to add strength to those membranes, especially in the case of eukaryotes that don't have cell walls to help protect them. All right, as far as the nucleus, it's going to have all of the DNA that makes up all the genes that encode the genetic information for that organism. And the way those genes are housed is they're housed in nucleosomes. What does that mean? For those of you who have had cell bio, what's a nucleosome? What's a histone? A protein. It binds DNA. That's right. Histones are actually positively charged proteins. They're attracted to the negatively charged sugar phosphate backbone of DNA. And they form this tight, compact structure known as a nucleosome with the DNA wrapped around the histone proteins. Any idea why this would be beneficial for the DNA to be wrapped around these proteins rather than just naked DNA in the nucleus? What is it?
Harder to damage.
It's harder to damage and degrade the DNA, right? You can't really access it as well in terms of all of those proteases or nucleases that could go in and try to damage these important macromolecules. So here they're better protected, the DNA is better protected, and the proteins, for that matter, are protected in this nucleosome complex. Now, as far as bacteria go, they don't have histones and nucleosomes wrap their DNA together with protein. But archaea do have histones and nucleosomes, which is interesting, right? And one of the key differences between between archaea and bacteria. So I'll just remind you, when we look at the tree of life, remember, archaea, even though they're prokaryotes, are more closely related to eukaryotes than they are to bacteria. And here is yet another example of how at the molecular level, we have a similarity between archaea and eukarya, both having these histones and nucleosomes. All right. The nucleus is also enclosed by a double membrane, not just a single one. And the two membranes are going to interact with the environment inside the nucleus and outside the nucleus. We can call that area inside the nucleus the nucleoplasm. And then the area, of course, inside the cell that's outside the nucleus would be your cytoplasm. So this nuclear membrane is going to need to be amphipathic. What does that mean? What is it again? Excellent. It's going to have to be a membrane made up of a lipid bilayer with both hydrophilic and hydrophobic parts. Remember the hydrophilic polar heads and the hydrophobic fatty acid tails? This is what we see surrounding all of these organelles, including the nucleus. And then within the nucleus, we have a special place known as the nucleolus, which is the site of ribosomal RNA synthesis. Ribosomal RNA being a major component of the ribosome. And why is the ribosome important? Protein synthesis occurs at the ribosome. Very good. Let's talk a bit about mitochondria and chloroplasts, because eukaryotic microbes have to have these organelles, and the reason is because they need to be able to produce energy, Okay, and these are the two organelles where that will happen. So mitochondria are called the powerhouses of the cell. They contain the site for cellular respiration, which will allow chemical energy in the form of food to be converted into usable energy that the cell can use. And then chloroplasts, of course, are the site of photosynthesis, where photosynthetic organisms, many of which are microorganisms, are able to use light energy to convert into chemical energy so that the cell can function. Now, there is a particular hypothesis out there, which is being supported by more and more and more data every day that passes, it seems, known as the endosymbiotic theory. I guess it's not until a later slide, so we'll get to that in a minute. But the endosymbiotic theory states that these particular organelles actually started as bacteria. So they have their evolutionary roots, we say, in bacteria. These guys were thought to be once free living organisms that functioned on their own without the need of anything else. And then they got basically enveloped by the first eukaryotic cells. And because they were so advantageous for those eukaryotic cells, they just kept them along. And every time that they made a new bacteria, the mitochondria would go along with it. So these particular organelles provide energy. We said mitochondria is the site of cellular respiration for aerobic eukaryotes, and of course chloroplasts contain chlorophyll necessary for photosynthesis. I'm sure a lot of you have had this before in your cell biology or general biology classes. So here's a picture of the mitochondria. The mitochondria has a really cool structure. It has two membranes, an outer membrane and then an inner membrane. And the inner membrane have all of these infoldings and invaginations to increase surface area. Why do you think it's important for the mitochondria to have more surface area in its inner membrane? Yes, one of the most important parts of the cellular respiration pathway is called the electron transport chain. And the proteins that are important in the electron transport chain are embedded in that inner membrane. So if you have more inner membrane available, you can have more electron transport, and therefore the cell can make more ATP. So that's why we have these invaginations called crystals. The number of mitochondria in a cell are going to vary. It can be very few to up to more than 1,000, believe it or not, within a cell. And as I said, those cristae, or those folded parts of the inner membrane, are super important to maximize surface area for proteins involved in cellular respiration. We call the part inside the cristae, inside that inner membrane, the matrix. That's the innermost area of the mitochondria, and it contains citric acid enzymes. And I love these pictures down here, taken with electron microscopy. Do you think it's TEM or FEM? TEM, absolutely. We're looking at a slice through the middle of the mitochondria almost. And you can see all these lines here in the middle are actually infoldings of the inner membrane, where you can have all those different proteins involved in the electron transport chain. Same here. Do you guys see all that? It looks like it's almost striped, right, like a zebra, but these are actually infoldings that connect with the inner membrane. So you can have so many more proteins embedded in those membranes than if it was just a straight inner membrane that mimics the outer membrane. Now, chloroplasts are similar to mitochondria in many ways. They also have a double membrane. And they're also involved in a very important metabolic process, but this time only for the photosynthetic organism. They have an inner membrane that surrounds stroma, and the stroma contain large amounts of a key enzyme for the Calvin cycle, which is a part of photosynthesis, so that sugars can be made from carbon dioxide. The thylakoids are going to be these flattened membrane discs that contain chlorophyll, and they contain the ATP synthetic component and form a proton motive force. So the stroma are to the chloroplast as the cristae are to the mitochondria, right? It's very important to have those membrane areas for various reasons. So here's that endosymbiotic hypothesis I was telling you about. So scientists now have quite a bit of evidence to suggest that mitochondria and chloroplasts were once free-living bacterial cells. Long time ago. Okay, we're talking about like origin of the earth time, or, well, origin of living things time, I should say. And that they descended from respiratory and phototropic bacterial cells, but then they became associated with eukaryote hosts at some point in early history, early Earth. And, you know, you may say, okay, that's fine and all, but where's the evidence for that? And the evidence includes the fact that mitochondria and chloroplast have that double membrane, right? The idea being that that outer membrane would actually be from the host cell when it sort of got engulfed by the host, by eukaryotic cell. Then it got pinched off and that gave it its double membrane. But also the fact that these organelles are the only organelles that have their own DNA. Okay, we know a cell has DNA, right? That's the genome. But in addition to the genome, there are mitochondria and chloroplasts also have their own DNA in there. And it's small and it's circular. So you may have heard of mitochondrial DNA, right? Which comes from your mom. That's what we're talking about here. So why in the world would an organelle, if it was always just an organelle, have its own DNA unless it was once a free-living organism? Eukarya are hypothesized to have originated from a symbiotic fusion of the host with the mitochondria. Specifically, an archaeal host and a mitochondrial endosymbiont. And then the eukaryotic host cell is thought to have acquired a chloroplast endosymbiont to become the ancestor of the phototropic eukaryotes that we know about today. And that's how some eukaryotes are thought to have developed the ability to do photosynthesis. It's pretty interesting, the articles if you read about them. And it's convincing too. All right, other eukaryotic cell structures that we see in eukaryotic microorganisms include the cytoskeleton. What's the point of a cytoskeleton? Structure, an internal skeleton, right, to provide structure. And this is super important too for organisms that don't have a cell wall to provide that kind of structure. So if they can provide it from the inside. So the cytoskeleton is made up of different kinds of fibers, if you will. There's the microtubules, which are hollow tubes that maintain the cell shape. They're also involved in motility and moving chromosomes and organelles. Then there are the microfilaments, the second type of fiber that makes up the cytoskeleton. These are made up of actin proteins. So you may also hear microfilaments called actin filaments. The same actin that works together with myosin in your muscle cells. It also maintains and changes the cell shape. It's also involved in motility, especially for amoeba and in cell division. And then lastly, the third type of fiber that I wanted you guys to be familiar with that makes up the cytoskeleton are the intermediate filaments. These are of intermediate diameter between the microtubules and the actin filaments. And these are going to be fibrous keratin proteins, the same keratin that gives strength to your hair and your nails. That's what we're talking about here. And it's important for maintaining cell shape as well and positioning organelles where they should be inside the cell. Let's talk about the remaining organelles that we haven't touched on yet. There's ER, endoplasmic reticulum. You guys know this already. It's a network of membranes, right? Some of the ER is attached to the outside of the nucleus, and some of it is sort of in the cytoplasm a bit. Which one is attached to the nuclear membrane? Y'all know? Rough ER. What makes it rough? Yeah, you got ribosomes attached to that one, and it's responsible for making proteins. And then the smoothie ER is actually responsible for making lipids, which are so important for the proper cell's function because it makes up the membrane, right? We need lipids for their lipid bilayer and for carbohydrate metabolism. The rough ER is going to make your proteins, specifically your glycoproteins and your membrane proteins. And then there's the Golgi. I remember being told from a biology teacher when I was like in middle school that the Golgi body is what we called it back then, now the Golgi complex. It's like the post office of the cell, right? Because it sort of packages it and tags it and sends it on its way, right? Because this is where the different, whatever is being modified, right, a protein, if you will, is going to be accepted by the Golgi, modified in whatever way it needs to be. Maybe a tag needs to be put on it in order to say a signal sequence that says, okay, this one's destined to be exported from the cell. This one's destined to go to the nucleus, right? That tagging, those signals are going to be put on at the Golgi. And then lysosomes, these are important because they have digestive enzymes inside of them. And what do you think needs to go there? Trash. Good. Take the garbage out. Yeah, if you have maybe a misfolded protein, that could be detrimental to the cell. That can lead to disease, right? Or maybe you have mutated DNA that's not properly replicated or transcribed. Or maybe a cellular waste product, right? Well, all that needs to head to the lysosome so that the digestive enzymes can chew it down like Pac-Man, right? And get all the good parts out of it. Get the good parts out of the mutated DNA, get the amino acids out of the misfolded protein, right? Get all those great atoms out that can be reused and then put into something that is functioning properly. So lysosomes are super important for cleanup. And then our friend flagella, which we started this lecture with, we also have them on some eukaryotic microorganisms, as well as cilia. But they're going to function in motility, right, and allow cells to move by swimming. The cilia are shorter flagella, and they kind of beat rhythmically in motion. And so they work together. They cover the whole surface of the microorganism and allow for it to move together. Whereas the flagella, we already know, are going to be at the poles typically. And there will only be one or a few of them. Eukaryotic flagella are long appendages. They propel through a whip-like motion. So this is an important distinction, guys. We now know, thanks to some really cool experiments, that bacteria move counterclockwise or clockwise. Well, this would be counter, this would be clockwise, right? And that's what gives the tumble and run. Well, eukaryotic flagella have a whip-like motion, like this. And I'm not exactly sure why they move differently. I don't know if it's known why they move differently, because both of them are super efficient at moving the cell from one location to another. But at any rate, eukaryotic flagella have this whip-like motion. Dynein is an important protein that's attached to the flagella and uses ATP to drive this motility. Say something?
So like archaea, it's a feature.
Yeah. Yeah. And they are structurally and functionally different than the prokaryotic flagella that we talked about.
Just to clarify, the archaea, do they move similarly?
The archaea, you know, that's a good question. I don't know the answer to that, but I can look it up and let you know. Yeah, I don't know if the archaea are rotating or whip-like. I believe they're rotating, but I'm not 100% sure, so I'll need to get back to you. Oh! Well, we got through about half of what I wanted to today. But hopefully you guys enjoyed the lecture, and I'll see you